Welcome back. In this web lecture, we're going to finish up our discussion about plant responses to internal and external signals. And so in the last web lecture, we talked primarily about how this information is transmitted through the plant using hormones to convert an external signal into an appropriate response to the environment. So in this web lecture, we're going to talk more about what kinds of things in the environment plants respond to, how they respond, and how hormones are used to coordinate these responses to the environment. So I just wanted to remind you first of this slide. You saw it in the last web lecture about information processing in plants. Remember that plants gather, process, and respond to the information they monitor in a three-step process. Sensing the environmental conditions, so sensory cells are going to receive an external signal and convert it somehow into an intracellular signal that can be conveyed through the body. The sensory cells then send a signal to target cells in other parts of the plant body, which are then going to receive that signal and change their activity in some way to produce an appropriate response. So we focused primarily on these last two steps in the last web lecture. Now we're going to think a little bit more about how plants sense their environment, how factors in their environment cause them to change in some way. And so the most important environmental factor that plants respond to is light. So light cues many key events in plant growth and development. And the effects of light on plant morphology are called photomorphogenesis. So these are changes in the plant's structure and its body in response to changes in light. Plants detect not only the presence of light, but also its direction, intensity, and wavelength. We saw a little bit about how plants detect the direction last time when we thought about phototropism. And as we saw way back when we were first looking at photosynthesis, this graph known as an action spectrum is a good way of depicting the relative response of a process to different wavelengths of light. So light is separated into its different wavelengths, different colors of light. Some process that depends on light is measured, and we can see which wavelengths of light are most important to creating whatever that physiological response is. So action spectra are useful in studying any process that depends on light. And action spectra can be used to determine which responses are mediated by a specific photoreceptor. So photoreceptors are going to be specific to particular wavelengths of light, and the action spectra can give us some clues as to what photoreceptors we should be looking at. So there are two major classes of light receptors: blue light photoreceptors and phytochromes. So let's first look at the blue light photoreceptors. So here we have an example of an action spectrum with some measure of the effectiveness of this process of, in this case, phototropism, and we see that it's maximal around a wavelength of 436 nanometers in the blue range. So blue light initiates a variety of plant responses such as hypocaudal elongation, stomatal opening, and also phototropism, what we looked at in the last web lecture. Phototropin is a protein kinase involved in mediating plant responses to blue light. Again, we saw this protein kinase in our last class, involved in this phototropic response of the plants bending toward the source of the light. So now let's take a look at these phytochrome photoreceptors. Phytochromes are pigments that regulate many of a plant's responses to light throughout its life. We saw them in the example of de-etiolation at the beginning of this chapter. So in addition to de-etiolation, these responses include seed germination and shade avoidance. Many seeds remain dormant until light and other conditions are near optimal. So remember that hormones are going to play a big role in keeping these seeds from germinating, causing them to remain dormant until conditions are right for them to grow and survive. In the 1930s, scientists at the U.S. Department of Agriculture determined the action spectrum for light-induced germination of lettuce seeds. So let's see what they found out. So it turned out that red light increased germination, while far red light, so this part of the red spectrum that's just at the very border of what we can see, this far red light inhibited germination. And they found that the effects of red and far red light are reversible. So whatever was the final kind of light that the plants were exposed to determines the response. So here you can see germination rate. This is from the paper where this was discovered. If red was the only light they were exposed to, 98% germination rate. If far red was the last one exposed to, so red and then far red, we get only about 54% germinating. If we go red, far red, red, 100% germination. Add a burst of far red to that, get 43. Put in another red after that, 98, and so on. So what's going to determine the germination rate is the last light they were exposed to. And the photoreceptors responsible for the opposing effects of red and far red light are receptors called phytochromes. So phytochrome is a red, far red receptor, and the same pigment absorbs both red and far red light, existing in two alternate shapes. So one shape absorbs red light, the other shape absorbs far red light only. And they'll switch between these two configurations depending on what light they were most recently exposed to. So such switching behavior is known as photoreversibility. So here we have an image of this. We've got one shape that's going to be able to absorb red light. This is the PR conformation. Once it absorbs red light, it's going to change its shape into the PFR conformation, the P far red. Now in this shape, it's only sensitive to far red light. So here it is in the PFR conformation. When it's hit by far red light, it changes back to the PR conformation. And now it's receptive to red light. So it works like a switch, depending on what light it was exposed to most recently. So PR, phytochrome red, absorbs red light. PFR, phytochrome far red, absorbs far red light. And this switching back and forth between these two conformations of the phytochromes plays a big role in shade avoidance. So remember that plants that are growing close to each other can be shaded out by other taller plants, and the trick is to be able to grow up above that other plant so that you can get to the sunlight. So the phytochrome system also provides the plant with information about the quality of light. So leaves in the canopy absorb red light. They do this for photosynthesis, and they allow far red light to pass through to the shaded plants below. So if you're under one of these trees and you're being shaded, you're getting less red light because it's absorbed by the leaves above you, and more far red light. So when that happens, when a tree is shaded, the phytochrome ratio shifts in favor of phytochrome red. So it's absorbing far red light and changing to the PR conformation. And that's going to induce vertical growth to try to get that tree or plant up higher than the tree or plant that's shading it. And this is called the shade avoidance response. The vertical growth, the growth in height, is the primary response to the presence of phytochrome red. And the presence of phytochrome far red has a bunch of different responses. So this is the form that phytochrome's in when it's getting lots of full sunlight, including red. So seed germination is going to be one of the responses to the far red version of this phytochrome. Inhibition of vertical growth and stimulation of branching. Remember, this trade-off between vertical growth means you don't get as many new leaves coming off in these horizontal branches. So when you're getting enough light, it makes more sense to invest in becoming bushier. It's also going to help to set internal clocks. So this resets those circadian rhythms in the morning. And as we're going to see, it's also going to control flowering. So let's take a look at the signals that promote flowering. So flower formation begins when an apical meristem stops making energy harvesting stems and leaves and begins to produce the modified stems and leaves that form flowers. So it's a switch over from one of these axillary buds forming stems and leaves, and instead they're going to form flowers. So the signals that promote flowering are based on the number of hours of light and dark in the day. And the switch that triggers flowering shows red, far red photoreversibility. To ensure flowering at a seasonally appropriate time, plants must be able to detect daily cycles to be able to detect the changing of the seasons through the year. So let's think about this idea of photoperiodism. Photoperiodism is any response by an organism that's based on photoperiod, the relative lengths of day and night. This is going to give the plants clues as to what season of the year is, what kind of temperatures to expect, and when it is an appropriate time to produce flowers. So in plants, the ability to measure photoperiod allows individuals to respond to seasonal changes in climate. So this allows plants to flower when pollinators are available and resources for producing seeds are abundant. Phytochrome conversion marks sunrise and sunset. The default state for the phytochrome is to be in this red configuration. Once the sun comes up, it's going to start converting that phytochrome red to phytochrome far red, and that's going to be what the plant's going to use to know that the sun has come up. Conversely, at the end of the day, those phytochrome far reds are going to start switching back to phytochrome reds to tell the plant that the day is over. So plants fall into three categories in terms of how they respond to these photoperiods. There are long day plants that bloom in midsummer when days are longer than a certain length. Short day plants bloom in the spring or fall when days are shorter than a certain length. And then there are day neutral plants that flower whenever they're ready without regard to photoperiods. So when they reach a certain age, maturity, they'll go ahead and flower, and it doesn't matter what time of year it is. And it turns out that it's not so much the length of the day that determines when these plants are going to flower. It turns out that what they're really detecting is the length of the night. So in the 1940s, researchers discovered that flowering and other responses to photoperiod are actually controlled by night length and not day length. We still use the terminology long day and short day plants by tradition, but this is really what's happening. So interrupting the night period with a light flash changes the flowering response, triggering the phytochrome switch. So if you've got a short day plant that needs a long period of dark in order to flower, if you turn on the lights in the middle of that dark period, you're going to disrupt this flowering response. We're also going to see this reversibility of the phytochromes in this response. So here we have an example of a long day plant or a short night plant, and a short day plant or a long night plant. So here we have a long night, so this is going to be fall or winter where the light nights are longer, and our short day plant is going to flower. Our long day plant will not. But if in the middle of that long night you produce a flash of red light, that's going to interrupt the light. There's not going to be the period of continuous dark that's needed by the short day plant to be able to flower. But this long day plant is going to detect that it's a shorter night because it was interrupted, so it was like 2 short nights instead of 1 long night, and so it's going to flower. The short day plant will not. If, however, you follow that up with a flash of far red, that's going to switch the phytochromes back over to the red configuration, and it will be like that flash of red light never even happened, and it's going to be responded to as if this was a long continual night again, and the short day plant will flower, the long day plant will not. And you can do this as many times as you want. It just depends on what was the last light they were exposed to, where that switch has been left. That's going to determine the flowering or not. So if we end with a flash of red light, the long day plant flowers. If we end with a flash of far red light, then the short day plant flowers. So this can be used to manipulate flowering time for people in the floriculture industry to be able to get flowers during times of year when they normally wouldn't flower. Some plants are going to flower after only a single exposure to the required photoperiod, so it just takes 1 long night or 1 short night to induce flowering. Other plants need several successive days of the required photoperiod to make sure that the right season is really coming. And then still others need some other kind of stimulus to help them to detect when is going to be exactly the right time to produce flowers. And one example is this phenomenon of vernalization, where plants have to be exposed to a prolonged period of cold, or a period of cold for a set length before they'll flower, because then they know that the right amount of time has passed for winter, and then now it's time for spring, and they can go ahead and produce flowers. So how do plants sense these changes in day or night length to initiate these chains of events leading to flowering? How do they know how long the day or night is? So plants do have these internal clocks, these circadian rhythms that are reset to a 24-hour clock each morning using these phytochromes switching from the red to the far red configuration. And the clock protein levels rise during the day and trigger the expression of a gene called constans, CO. The constans protein is a transcription factor that affects the production of a flowering hormone. So however long the light lasts during the day, these constans proteins are going to accumulate. So longer days, you'd end up with a larger amount of this constans protein. For shorter days, you're going to end up with a smaller amount of this constans protein at the end of the day. And the effect of the constans protein varies. So in long day plants, high levels stimulate flowering. In short day plants, high levels inhibit flowering. So it's the same signal doing the same thing, but the mutation that occurred, or the change between these two kinds of plants, is in how the plants respond to the high levels of this protein. So this leads us to believe that there's some kind of hormone involved in initiating the production of flowers. So experiments have shown that exposing even one leaf on a plant to the conditions necessary to induce flowering may result in the whole plant flowering. And so this is a similar but not identical experiment to the one that you saw in your book. So this leaf, which has been exposed to the right photoperiod for flowering, is grafted onto this plant that was not exposed to the right conditions for flowering, is going to go ahead and produce flowers based on some kind of signal from this leaf. And the flowering hormone is named florigen. So the gene flowering locus T promotes flowering when activated. So for a long time it thought that this flowering hormone must be some kind of small mobile molecule, and that's pretty much what people were looking for. And then the gene flowering locus T was discovered that has the action of promoting flowering when it's activated. So we discovered this through mutant studies. And so it turns out that this flowering hormone is actually a protein. So the protein product of the FT gene is transported from leaves to the shoot apical meristem, triggering the activation of genes required to convert the stem into a flower. So it's not just light that plants respond to. There are a large number of things that they're going to need to respond to in their environment. And because plants can't get up and move around and take direct actions on their environment, plants must adjust to a range of environmental circumstances, and they need to do this through developmental and physiological mechanisms. They need to change something about themselves to be able to adjust to these environmental conditions. So let's first take a look at how plants respond to gravity. So obviously plants need to grow in such a way that their roots grow down into the soil, their stems grow up toward the light to collect sunlight. So they have this phenomenon called gravitropism, or movement in response to gravity. So gravitropism is the ability to move or grow in response to gravitational pull. So if they are moving or growing in the same direction that gravity's pulling, that's called positive gravitropism. So roots respond to gravity by growing down. Shoots exhibit negative gravitropism, growing in the opposite direction from the gravitational pull, and they respond to gravity by growing up away from it. And it turns out that the ends of root tips have the sensors that are going to detect this gravitational pull, and they're going to be located in this protective root cap that we talked about before. That's a layer that surrounds and protects the apical meristem of the root. So these were more experiments done by Darwin and his son, and they found that a root stops responding to gravity if the root cap is removed. So there's something going on in this root cap that's what is allowing the plant to sense and respond to this gravitational signal. So gravity sensing occurs in cells containing starch granules, and they're located in the center of the root cap. So here we see this population of cells with these starch granules that are all kind of collected at the basal part of the cell. This gives rise to what's known as the statolith hypothesis. And this is the leading explanation for how plants sense gravity. And it's based on two interconnected ideas. First of all, ameloplasts, which are dense starch-storing organelles, respond to gravity by being pulled to the bottom of root cap cells. So these have a greater density than the rest of the cell contents, and they're going to tend to be pulled down by gravity to the bottom of the cell, whichever is the bottom with respect to gravity. And then the position of these ameloplasts activates some kind of sensory proteins that are able to detect that weight on top of them. And this is what's going to initiate that gravitropic response. So these activated pressure sensors, so if this root was growing sideways, they would be pushing on the side and cause this gravitropic response. And it's also thought that auxin is the primary gravitropic signal. So a series of events results when root cap cells detect changes in the direction of gravitational pull. For example, in that photo you saw where the plant was laid down on its side in its pot. So normally, auxin flows down the middle of the root and then toward the perimeter and away from the root cap. So here's a normal flow of auxin when the root is oriented appropriately with respect to gravity. If the root is tipped, so in this case we're seeing it tipped completely sideways, sensory receptors are going to trigger changes in the position of auxin transport proteins. So they're going to transport auxin in a different direction, same thing that we saw in the phototropic response. The lower portion of the root is going to receive more auxin while the upper portion receives less. And so this is the tricky thing that you need to remember, is that these hormones are going to have different effects in roots than they do in stems. And a lot of times it will be the exact opposite effect. So in the case of roots, high auxin levels inhibit growth, whereas low auxin levels promote elongation. The upper portion that's getting less auxin is going to elongate, the other side is going to shorten, causing that root to bend downward. So now let's take a look at how plants respond to mechanical stimulus, so being pushed on by wind or physical touch. So plants respond to mechanical stresses such as wind and touch, and growth responses are going to produce stout, stiff stems, so sturdy, short, thick stems that are going to be more resistant to mechanical pressure. Directional growth will move plants either toward or away from contact with an object as necessary. So sometimes if a plant is, for example, a vine and wants to grab onto that object and climb up it, it's going to grow toward that object. If it's a plant that needs space, wants to get away from things to get maximum sunlight, that might cause that plant to move away from the object. And then in other kinds of plants, such as a Venus flytrap, these mechanical stimulations might create a quick movement, causing the Venus flytrap to snap shut when it senses the pressure of that fly on its sensors. So a large suite of genes may be transcribed in response to touch or other mechanical stimuli, and the protein products of these genes are going to act to stiffen cell walls to make the cells sturdier, resulting in shorter and stockier plants. So the term that we use to describe this change in shape and form as a response to mechanical stimulation is thigmomorphogenesis. So that refers to changes in form that result from mechanical disturbance, touching, pushing. And for example, rubbing the stems of young plants a couple times daily results in plants that are shorter than the controls, so these guys here. So thigmotropism is growth in response to touch, so movement in response to touch. And it occurs in vines and other climbing plants. So when vines detect something that they want to climb on, they can send out these tendrils that are going to coil around their support and help them to grow up into the light. Some plants undergo rapid leaf movements and respond to mechanical stimulation. So for example, these touch-me-not plants, Mimosa pudica folds its leaflets and collapses in response to touch. And this actually can happen throughout the plant by just touching one little leaflet. So that signal is actually communicated at some distance from the source of that pressure. And this touch response that can move through the plant and be communicated relatively quickly results from the transmission of electrical impulses called action potential. So this is similar in many ways to the way nerve transmission works in animals. So plants also need to respond to environmental stresses, so things that are going to be detrimental to their survival and growth in the environment. So environmental stresses have a potentially adverse effect on survival, growth, and reproduction. And these stresses can be biotic as a result of other living organisms, or abiotic, part of their non-living environment. So biotic stresses can include herbivores and pathogens, and abiotic stresses include drought, flooding, salt stress, heat stress, and cold stress. So we're going to focus in this web lecture just on the abiotic stresses. So let's start with drought. We've already thought a little bit about how plants respond to water loss. So plants may wilt or die when water loss by transpiration exceeds water absorption. And as we've seen, during drought, plants reduce transpiration by closing stomata, reducing exposed surface area, and in some species, just shedding the leaves entirely to be able to conserve that water. If we think about flooding, so waterlogged soils can also be very bad for plants. Remember, these are still undergoing aerobic metabolism using oxygen. These are eukaryotic organisms with mitochondria that have these aerobic pathways. So waterlogged soils lack the air spaces needed to provide oxygen for cellular respiration in the roots. So one of the ways that some plants deal with this is by the enzymatic destruction of root cortex cells, creating air tubes that work kind of like a snorkel to be able to get air down into the roots through the root tissue itself. So this is going to help plants survive oxygen deprivation during flooding for at least a short period of time. And some plants, such as mangroves that are perpetually in waterlogged locations, also produce aerial roots that are going to be able to transmit the oxygen down into the submerged regions of the roots. So here's a micrograph showing the difference in this control root that's in an aerated soil that has sort of normal cortex cells around the perimeter, around this vascular cylinder. And here we have an experimental root that's in flooded soil that has broken down a lot of these parenchyma cells in the cortex and created these air tubes, these air spaces to get oxygen down into the roots of the plant. Another problem for plants can be salt stress. So sodium and some other ions can be toxic to plants in high concentrations. And one of the big problems is that salt in the soil can lower the water potential of the soil solution and reduce water uptake or even draw water out of the root cells, which is a very, very bad situation for the plants. So plants respond to salt stress by producing solutes, so organic molecules that are going to be dissolved in the tissues, decreasing that solute potential. But these are going to be solutes that are tolerated at high concentrations. They're going to keep water flowing into the plant tissue, but these are not going to be materials that are toxic to the plant. So this process keeps the water potential of cells more negative than that of the soil solution, and will keep that plant from having water drawn out of it and becoming dehydrated. Another problem for plants can be heat stress. So as we know, excessive heat denatures proteins, and proteins in plants are no exception. So one thing that all plants do is evaporative cooling through transpiration. So transpiration has a dual function of moving materials through the vascular system of plants, moving water and materials, but it also helps in thermoregulation of the plant, keeping the plant cool by evaporative cooling. In very, very dire situations where this evaporative cooling is not enough and the temperatures become very high, plants can produce proteins called heat shock proteins that can help and protect other proteins from becoming denatured, either by giving them a scaffold that allows them to refold into their correct shapes. But these are kind of short-term solutions that can help a plant survive a short period of heat stress. And finally, something that can be very damaging to plants potentially is cold stress. So cold temperatures, remember, decrease the membrane fluidity. We need this membrane to be mobile and fluid for the membrane proteins to be able to move around and get where they need to go to perform their functions. And so we've seen already when we looked at cell membranes some of the mechanisms any cell can use to be able to deal with cold temperatures. So altering the lipid composition of membranes is something that can help with this membrane fluidity. And they're going to alter these membranes by adding more unsaturated lipids to the membrane to space out those lipids in the phospholipid bilayer and contribute more to fluidity. So another problem is that the formation of ice during freezing reduces the water potential outside the cell. So basically it is decreasing the amount of liquid water available. So that's going to decrease the solute potential because there's going to be the same amount of solutes and a lot less liquid water. So cells of many frost-tolerant species will do exactly what we talked about in salt stress. They're going to increase the solute concentration by producing organic molecules as solutes, and that's going to reduce the water loss from the cell by decreasing also its solute potential. And plants, as well as many other organisms, can produce antifreeze proteins that are going to help to keep ice crystals from growing within their cells. So it's going to hinder the formation of ice crystals within their own cells to keep their cells from freezing up. Also, frost tolerance can be increased in some crop plants by engineering these antifreeze genes into their genomes. So crops that normally wouldn't have the capacity because they don't normally grow in places where there's frost, they can be genetically engineered to produce these antifreeze proteins to help them get through the winter. So in this web lecture, we've seen some general mechanisms that plants use to respond to their environment. So we saw several ways in which plants can alter the timing of life events, from germination to flowering times, in response to what they're sensing in their environment. Another way is growing in ways that adjust to the environment. So if they're being shaded, they may grow vertically up out of the shade. If they're growing in the wrong orientation with respect to the ground, they can change their orientation in response to gravity. And they can alter the structure of their tissues. So as we saw, the breaking down of those cortex cells to form these air tubes in flooding conditions. But they can also alter the chemistry of their cells, changing the solute concentration from within the plant itself to be able to deal with changes in solute potential, either due to salt stress or cold stress. They can produce specific proteins that are going to help with dealing with heat or dealing with cold. So these are all mechanisms that sort of happen within the plant. The plant is changing itself in some way to deal with its environment. And this section marks the end of the material for this exam, but also the section on plants. So as we move forward in the next class and beyond the exam, we're going to be thinking about organisms that respond to their environment in a very different way. So we're going to be thinking about animals and how they can actively respond to their environment. So we're going to start with animal organization and move up through different animal systems leading to their response to the environment.